Fish that live mainly or completely in freshwater habitats such as lakes, rivers, ponds and streams with salinities below 0.05% are known as freshwater fish. From fast-moving mountain streams to the placid water of lakes and wetlands, these environments are diverse. The variety of location and distinctive adaptation that fish have evolved over time are responsible for this amazing diversity. In addition, freshwater fish are species that spend their entire life in either freshwater inland or brackish estuaries. Even in protected areas, there are still scarcity of information on Malaysia's freshwater ecology and its aquatic life, especially freshwater fish. Effective conservation and management plans require a thorough understanding of freshwater fish composition, distribution along river gradients, and relationship between environmental factors. This study examines the scale structure of a few selected cyprinid species found in the tributaries of Jengka River, which is Labiobarbus leptochilus, C. apogon, Barbonimus schwanenfeldi, Mystaculus obsutiratis, and Ostichilus vittatus. All these scale traits provide essential biological indicators of adaptation to specific freshwater environments. The overall shape, anterior, posterior, radi, and circular part of each species are thoroughly inspected. The study will further knowledge on cyprinid biology, taxonomy, and their interaction with the distinctive aquatic environment of the Jengka River tributaries by examining these characteristics. The result can also be used to set baseline data for upcoming ecological and conservation research in the area. Next, we move to the objective of this study. The objective is to determine the structural features of scales among selected cyprinid species inhabiting the tributaries of Jengka River in Felda, Semarak, Jengka 15, Pahang, and to compare the morphologies of scale structure of different cyprinid species in the tributaries of Jengka River. So, the significance of this study is this study provides important information about the condition of local fish population and the freshwater ecosystem by observing the morphological characteristics of the fish scales belonging to the fish-inhabited Jengka River tributaries in Felda, Semarak, Jengka 15. Study on scale structures is crucial as it provides advanced knowledge of the aquatic fauna in this area by offering vital insight into the taxonomy of the scale structure. The study will improve the methods of species identification by characterizing scale structure and demonstrating the influence of environmental conditions on this structure. Scale morphology is widely employed in study for growth analysis, age estimation, and taxonomy categorization. Accurate morphometric measurement of scale length, width, and distance between circuli are made possible by tools like ImageJ software, which enables researchers to record characteristics unique to a species. In comparison with SEM, ImageJ-based technique use optical pictures captured by combined microscopy to extract numerical information about size and shape. So, the finding of this study, in this study, I examined the scale structure of 5 cyprinid fish species found in the tributaries of the Jengka River, Pahang. All the species had cycloid scales, but each show unique characteristics. Labiobarbus leptochilus had strong anterior dominance with a swirl-like focus, while Ostichilus vittatus showed the most difference with a longer posterior field and 2 scale types. Cyclotellitis apogon had disc-shaped scales with symmetrical radii. Mystaculus obsutiratis had hexagonal scales with longer radii toward the back, and Barbonimus schwanenfeldi had polygonal scale with radii only in the front. The statistical analysis proved that Ostichilus vittatus was the most distinguishable species based on morphometric values. Overall, this study showed that scale structure can help identify fish species and reflect how they adapt to their environment. For scale measurement like L1 per L, L2 per L, L1 per L2, and width per L, when compared to the other species, Ostichilus vittatus display distinct variation in the majority of the scale measurement values. Unlike other cyprinids, it has scales with longer posterior portion. The focus was more to the front and the back portion of the scale was longer, as indicated by the L1 per L value being the lowest and the L2 per L value being the greatest. Significant species distinction was achieved using morphometric analysis using ImageJ and statistical comparison using Tukey HSV, with Ostichilus vittatus being the most morphological distinct. All things considered, the study showed that morphometric indices and scale morphologies are reliable, non-destructive method for identifying species and represent ecological adaptation within the Cyprinidae family. So, in conclusion, 5 cyprinid species which are Labiobarbarus leptochilus, C. apogon, Barbonimus schwennefeldi, Mystacilus obtuserotris, and Ostachilus vitatus collect from the Jengka River tributaries were successfully used in this study to characterize the morphological and morphometric features of cycloid scale. Based on shape, ready arrangement, focus position, circular density, each species in Cyprinid had separate scale features, indeed that functioning in differentiating each species. Labiobarbarus leptochilus show a groove anterior with a swirl-like focus very obviously at the front of the scale. Ostachilus vitatus had unique type of groove with two known structures of scale and its radii were conserved to be expanded more to the posterior side. C. apogon had disc-shaped scales with symmetrical paired radii extending across all areas of the scale. Mystacilus obtuserotris had hexagonal scales with longer radii showing to the posterior field. Meanwhile, Barbonimus schwennefeldi had polygonal scales.